Good morning, everyone, and welcome to today's webinar. We welcome you to the Algal Bloom Action Teams webinar series. This is our first webinar of the year, and we're glad you can join us. My name is Erica Kleitz, and I'm an Extension Educator with Michigan Sea Grant Extension, and I'll be your moderator for today's webinar. Today's webinar will feature two presentations focusing on the mysteries and secrets of harmful algal blooms. We'll start with a presentation from Dr. Barry Rosen on freshwater cyanobacteria before hearing from Jennifer Wisecaver, who will discuss the cryptic species that is Prymnesium parvum. All right, so this webinar, as I mentioned, is organized by the Algal Bloom Action Team. That's a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resources Research Institutes, the North Central Region Water Network, and University Extension within each state in the North Central region. In addition to this bi-monthly webinar series, we host an annual virtual symposium, and we're developing a website of resources that can be used freely for outreach and education around harmful algal blooms in the North Central region. I encourage each of you to visit our website, northcentralwater.org/habs, which we are putting in the chat now, and there you can explore some of our resources that we have to date, including a What You Should Know fact sheet, a fact sheet on preventing and treating HABs for landowners, and a HABs Frequently Asked Questions database. We're also excited to announce that we recently released a fact sheet focusing on HABs and their health-related effects on animals. This fact sheet includes information on the signs of toxicity in animals, tips for domestic animals and livestock, and an overview of HAB toxins and their symptoms. It also includes the contact information for HAB poisoning for each of the 12 North Central region states. You can find that fact sheet and others on our website. We will also be posting a recording of today's webinar on our website for your reference. Please note that we'll allow time for Q&A after both presentations. So to have your questions addressed, please post them in the Q&A panel, which is located at the bottom of your Zoom screen. If there's a question already posed in the Q&A panel and you have the same question, you can use the upvote function to help ensure we get answers to the most popular questions during the panel. Please note the Q&A panel function is separate from the chat function in Zoom. So if you can put your questions in that Q&A panel, that ensures that I'll see them when we get to that section of the talk. If you're having any technical difficulties during today's webinar, please let us know via the chat function, and we'll assist you as the best we can. You can also use the chat function to introduce yourself or discuss the research findings with fellow water professionals during today's webinar. All right, we're going to go ahead and get started here. Joining us first is Barry Rosen, and he'll be speaking about unraveling the secrets of freshwater cyanobacteria blooms. Dr. Rosen began studying algae during his junior year of college at the University of Connecticut. Algae have been his area of expertise ever since, through master's in Minnesota and PhD in Bowling Green, Ohio. He has worked in various universities around the country, the private sector, state and federal government, and finally in Southwest Florida at Florida Gulf Coast University. Through the network of colleagues and new contacts, he receives live samples for identification, culturing, toxin testing, and molecular techniques. He recently completed a photographic collection of 1,100 microscope images covering 300 genera to aid folks in the proper identification of these organisms. So I'm now going to stop sharing my screen so I can turn it over to Dr. Rosen.
Thank you. So we're good to go. Got the screen.
Yep, looks great.
So today I'm going to talk about some of the things we as people think about harmful algal blooms, and knowing that they have many more secrets that we're just not aware of. As a group of organisms, they've been around at least 3.5 billion years. And we put on our human glasses and say, well, why are they there and how are they being successful? So I'm going to talk about my thoughts on this, I'm an algal physiologist, physiological ecologist, and I've worked on blooms for a long time. So I have a great title. People in academia go, how did you get to be a world-class scholar? And it's very easy. You just get old doing this stuff. Pardon my humor. So in Florida, we have typical water bodies. They all look like this, and that's why people flock to Florida, especially the elderly who want to be exposed to toxins. But in reality, this is a typical bloom that we find in Florida in the early to mid-summertime. And blooms like this can cause ecological disruption, and it's mostly from hypoxia. We do not find that the organisms, although they may be producing toxins, they typically aren't the cause of the disruption of the ecosystem. They can also have taste and odor problems, and some find this aesthetically displeasing. I think it's a rather pretty color. So we start with the paradox of the plankton. Plankton is both eukaryotic and prokaryotic organisms, all living in a water body that has, zooplankton in it, has fish in it, and something's going on to have us have a very diverse suite of organisms all at once in the same water body, which you would think has the same nutrients and the same everything else. But understand that these organisms each individually have their own special properties for pulling up nutrients, adapting to light, to temperature, all of which makes it much more complicated than rocket science. So my question is...
Sorry to interrupt. Did you advance the slides at all yet? Because we aren't seeing them advance on our end.
Oh, I know why. Why don't I got it. Thank you for interrupting. So it's over here. No, it's not over there. Thanks for the question. So I'm not sharing the right screen is probably what it is. Advancing?
Yes.
All right. So this is the typical Florida picture. So my joke got ruined, but you can see that this is what we have to deal with down here. So the concept of paradox of the plankton, again, you've got this vast array of both eukaryotic, prokaryotic organisms living in the water. Some are rare, some are common, and they're each adapted to their own particular conditions. That's the way to put it. And the question you're all asking, how do you go from an organism that might be rare or just in the plankton to becoming a dominant? And you have quiescent species that become common and then dominate that water body. And how that happens is very, very complex. And I'm going to talk to you about some of the strategies that I've observed on what these organisms do. So they each have their own efficiency for every single element, macronutrient, micronutrient. They each have their efficiencies for each light intensity, light quality. They each have their own efficiencies for everything else that they might factor into their growth. And on top of that, you've got that daily changes, you've got circulation, you've got weekly, monthly, seasonal fluctuations, all affecting these factors. And that's why it's more complicated than rocket science, because you have all these organisms doing their own thing, responding their own way. But again, how do you get cyanobacteria to be the dominant? Well, first of all, again, these are cyanobacteria. And I say that because a lot of people say, oh, blue-green algae and algae are algae. Well, the only thing unifying all the algae is that they have chlorophyll A. As a bacteria, they have a cell wall. In this particular group, if you remember back to microbiology, if you had it, they have either gram-positive or gram-negative cell walls. Well, cyanobacteria have a gram-negative cell wall. And so you see that structure. This is a TEM image on the right that I took. On the left is a schematic of that, the layers that are involved in that. And that's very important to their survival strategy, having that gram-negative cell wall. Again, the unifying feature of all algae and cyanobacteria, algae is really a layman's term for all these organisms. They have chlorophyll A, that unifies them. Cyanobacteria only have chlorophyll A compared to some of the other chlorophylls. But here's the nest, the tree of life. And animals are on this tree, but everything that's a colored box is a kind of algae because it has chlorophyll A. So you have the green algae and the charophytes over on the right, cyanobacteria on the far left, but then you've got the euglenoids, the dinoflagellates, the chrysophytes, and the cyanophytes. All of them are that family, and there's red algae and brown algae. So as prokaryotes, there's no membrane-bound organelles, no chloroplast, no nucleus, nothing that's membrane-bound. However, they do have DNA, and they do have ribosomes, but they're simple organisms. They simply divide in half. No sexual reproduction, no motility by flagellum that we would have in other organisms. The number one job of a cyanobacteria, in my mind, again, I'm putting human traits onto this, is to capture sunlight. They have to capture sunlight because they're photosynthetic, and they can pull in, simple organic compounds, inorganic organic compounds like amino acids and sugars, but for the most part, they're photosynthetic. So they have this pigment array in a phycobilisome, that structure on the right, which includes the pigments you know about, the phycocyanin, which is cyan, your cyan, blue-green color. That organism on the left, full of phycocyanin, it looks blue-green. They have phycoerythrin, which is like erythrocytes, that reddish color. And that combination of the red and the green and all the other pigments that they have make them have a brownish color or a black color, or all shades in between. It's those pigments that allow them to collect through that antenna and pump all the collection of photons to the reaction center, which is the chlorophyll A. So what does those pigments do that allows them, and this is one of their strategies, to pick up wavelengths that the other organisms that don't have phycocyanin, that don't have phycoerythrin, they can't collect those wavelengths. They can't, they go right by the antenna. Whereas the cyanobacteria, they can collect the phycocyanin, the phycoerythrin. And again, with those other pigments, they have a very nice array to capture sunlight throughout the water column. Note also that cyanobacteria have chlorophyll D, which is in the infrared region. So as they do photosynthesis, again, number one is capturing light. So they want to make an ATP, which is, the energy currency inside the cell, and NADPH. They have a waste product called oxygen, kind of important to the rest of life, but that oxygen is really important to other species other than the cyanobacteria. They need oxygen to metabolize the sugars they fix, but it is, during photosynthesis, it is a waste product. And eventually when they split carbon dioxide and water, they make sugars. And again, that's a generic term. And they keep their pigments, by the way, in thylakoids. Again, not a chloroplast, but thylakoids. That's where the chlorophyll membrane and that array is embedded in that. So to be equipped to maximize CO2 capture, they use rubisco, ribulose bisphosphate carboxylase, the most abundant enzyme in the world. And to be prepared to handle the massive amount of CO2 that might be available, they have a carboxysome. It's an array of multiple copies of rubisco ready to be turned on and used as an enzyme to capture carbon dioxide. So you know about light. The red light doesn't penetrate as deep because the wavelengths are longer, and the blue light penetrates deeper. So this is an important thing to understand, that in that water column, the deeper the organisms are, if you can pick up these different wavelengths, you're going to be more successful. And this is true for all the algae, not just the cyanobacteria. So how do they get themselves up? Many of the cyanobacteria that are planktonic, so I say selected genera because not all the planktonic organisms have it, but many of them do, have a gas vesicle, not a vacuole. Vacuole would mean membrane bound. This is a series of proteinaceous structures, all made of the same protein, that fill with air and makes these organisms buoyant. This is a filamentous cyanobacteria, TEM on the left, and that big orange structure is the gas vesicle, fills with air. And during the day, as they photosynthesize and they make that sugar molecule bigger and bigger and add chains onto it, it collapses the gas vesicle. And when it collapses, the gas vesicle and the whole organism can float back down in the water column. And as it metabolizes the sugars at night, the gas vesicle can fill back up again and bring these organisms back up to the surface during the day. So that gives them a tremendous advantage. When they come to the top, they can shade out other organisms and they can get the maximum amount of photosynthesis that they need. And that CO2, C6H12O6 is ballast. Oh, by the way, when they go down in the water column and you have the thermocline down there releasing nutrients, they can pick up a load of nutrients when they're down there. This regulation has been evolved over millennia to make them a successful organism moving up and down in the water column. You can see the gas vesicles. They're also called aerotopes. If you look at this image here, you can see these bright, shiny objects inside the cell. This is a dilocospermum, and those are the gas vesicles. Note also these cells here, the heterocytes, are clear. There's no gas vesicles in them, and I'll talk about why that happens later. So this is a little schematic I did, and to know about when they're floating up, and let's say you had an initial distribution of 100,000 cells per liter, and there's about 20 micrograms of toxin per liter. That buoyancy could cause a big increase up at the surface of toxins and cells. And when the wind blows, they're very dynamic. They can move all around the lake in a heartbeat. And you can see that if you have a drinking water supply or people on the edge where they're recreating, they can be exposed to a much greater number of cells and their toxin load. Simple division, they simply divide, but that doubling is interesting because they grow fast. They're thermophilic. The warmer the weather, the more they like it. And we call it doublings. How many doublings per day? And in culture, I have grown these organisms growing 3 doublings per day, which means 1 organism first thing in the morning, then 2, then 4, then 8. That 3 doublings leaves a lot of organisms. And if you're trying to control these, if you don't kill them all, you've not done anything but leave more nutrients around for the outside the kill zone, as I call it. The caveat on this kind of fast growth is everything has to be just right. Nothing is limiting. So go back to that first diagram. You have the light, the temperature, the nutrients, everything that you need. Other strategies, you have organisms that stay up in the water column by just having a big morphology to their colony, like this Gliotrichia. If you ask for Gliotrichia in Wikipedia, this organism, this photograph I took while USGS, shows up, and a lot of people like it. I think it's a floating pincushion. This one has both aerotopes and its morphology. Little microeddies can keep it floating up in the water column, just like these two Dilocospermum. This is a 3D image that I took, and yeah, a little microeddy will pull these things back up in the water column. And then if you're a Phanozomenon floss aqua, AFA, you can form a big flake. If you see flakes on your water body, green flakes that look like lawn clippings, most likely AFA. And again, the morphology plus aerotopes keep this up in the water column. Many of the cyanobacteria that have heterocytes, especially only in the group the Nostocales. So the heterocyte, think about this. This is a simple organism. You think, oh, it's a prokaryote. Well, there's one specialized cell, the heterocyte, that can fix N2, nitrogen, out of the air. Nitrogen with its triple bond. There's 60 enzymes in the nitrogenase complex inside that cell. The only thing is that cell is inhibited by oxygen. So they go through extra special protections and they convert vegetative cells into these heterocytes by A, putting a lipid layer around the outside, and B, doing other things to really cut down on oxygen or preventing oxygen from getting into those cells. So here's a TEM of that lipid layer. It shows here around the outside, and you see these little plugs at the end on the light microscope. There it is right there. And this is a picture from my colleague Hans Perl, and he shows a TEM. And look at the bacteria. Those are heterotrophic bacteria on the heterocyte, which looks smoother. They're there taking any kind of leaking nitrogen coming out of the cell. Visible changes happen to the heterocytes. That's why they're easy to see. And there's the plugs you can see on the end there. They shut down the oxygenic part of photosynthesis, cut down and cut off photosystem too. So they aren't making oxygen, but they need the ATP. It takes 16 ATPs to break one dinitrogen molecule and get nitrogen transported into glutamate and then into amino acids. Once they make that nitrogen, it has to get transported across that plug into the adjacent cells. So like plants, instead of a plasmodesmata, these are little microscopic, small plasmodesmata for transporting the fixed nitrogen through the lipid layer, through the adjacent cells. They'll turn on more heterocytes when the water body is nitrogen limited, and nitrogen fixing cyanobacteria, which are probably present in your water body, will start doing better when nitrogen becomes limiting. Another trick of the trade. Get more CO2 into the cell in whatever form they want to use it. There's active transport, there's passive transport, because Rubisco, that enzyme inside the cell, even though it's there ready to grab CO2, it can accidentally grab oxygen and cause photorespiration. And it's not the right products that it would make. It's not going to make the right kind of sugars. So if you can increase your CO2 level inside the cells up to 1,000-fold, Rubisco won't make the mistakes. So that is a mechanism that's pretty cool that a lot of you may not have heard about. So 5 separate transport mechanisms, some requiring ATP. So I knew to keep this short. I don't know how much time I've taken, but one of the interesting things is, and this blows anyone that's an engineer's mind, is that colonies may look perfectly healthy under the microscope, but know that every blue cell in here is dead. And second of all, the live healthy cells are not going to release toxin because it is a very large molecule. It's very expensive to make. There's a number of metabolic pathways that it serves. It's not about killing other organisms. We really shouldn't call it a toxin. It's toxic to, mammals and maybe some other organisms, but they evolved this toxin 2.1 billion years ago. So it's really a metabolic secondary metabolite that's important to the cells. You wouldn't keep a pathway around that's this expensive unless it served multiple purposes. So even though we are measuring toxin, when we have to do it, we actually have to smash the cells, freeze thaw techniques, sonicate them, whatever you do to get the toxins out. And by the way, the pink cells on here are actually green algae, which are hitchhiking on this colony of microcystis because they want to get moved around in the water column too. And why not take a ride on something that's going to move up and down? When I take these images, I use a wide blue excitation. This is epifluorescent, and you do wide blue excitation, which allows you to see that chlorophyll fairly nicely, but it also shows you cells that are dead. And then it emits at a longer wavelength. Again, all the cells in a colony are not in the same physiological state. The most important take home on this is that when you go to a water body, the lifeguard isn't there, the bacteriologist isn't there, the microbiologist isn't there, and I inserted the word phycologist because I'm likely not going to be there. And as a phycologist, I study the algae. That's what phycology is, the study of algae. And a long time, my parents thought I was a psychologist. And I said, I do study the psychology of algae. Anyway, swim at your own risk. And know this, that cyanobacteria are art. So this is a project that Keith Lofton and I did on techniques for rupturing cells to look at toxins. This is a boiled gliotrichia, boiled for 15 minutes. Red means it's still intact. Green means the DNA stain I use lit up the bacteria that are on all the mucilage. And in the center of this colony, the bright yellow in there, that's where the heterocytes are. Look how strong this organism is to survive boiling and not allow its DNA to be stained. It's that mucilage, that extracellular polysaccharide. I didn't feel like I had enough time to explain all of that. And if you don't think there are, look at this gliotrichia from the Everglades. Basal cells that are blue, that are heterocytes. Then you have this big aconite-like structure. Then that yellow cell that's next is like a separation disc. Then another heterocyte that's red. And then a long 1.5 millimeter long filament that tells you that you're in the genus Gliotrichia. Actually, I had to wait for the Czech Republic to tell me that it wasn't Rivularia. One last slide. This is who I am. And in case you don't know it, I collect live samples from around the country. And many of you have sent me samples. And the book that we talked about is now at the printer. It's 1,125 images, most of which were taken while I've been here. It also has across at least 10 divisions, and it's large format. It's 8.5 by 11 laid horizontally. It's spiral bound. It's glossy paper, and eventually I will have it in my hands and use it for teaching and working on a publisher. AWWA might publish it and have it for distribution. All right. I don't know how you're answering questions, but be glad to take them whenever they're ready.
Thank you. We do have some questions, but we're going to hold those for after the second talk. So you got lots of applause and appreciation for your images and that you shared today. So thank you for that. Let me share the screen to introduce. 1 second here.
So,
are you seeing my slide?
yep.
Okay, great. Okay, so our next speaker will be Jen Wisecaver, talking about extreme diversity and cryptic species in the toxic bloom-forming alga Prymnesium parvum. Jen Wisecaver is an associate professor at Purdue University in the Department of Biochemistry and the Center for Plant Biology. Prior to her current position, she was an NSF Plant Genome Research Program postdoctoral fellow at Vanderbilt University and earned her PhD at the University of Arizona in the Department of Ecology and Evolutionary Biology. Research in the Wisecaver lab is on genome evolution in eukaryotes. Currently, they are investigating how eco-evolutionary feedback shapes genome diversity and toxin production in Prymnesium parvum, one of the most problematic species of toxic algae in the country. All right, I'm going to stop sharing. Okay. And turn it over to Dr. Wisecaver.
Can you guys see my screen?
Yep, looks good.
Okay, great. All right, well, thank you so much for the invite. And it's great to see so many people online tuning in for a talk about toxic algae. And I have to thank Barry for setting me up perfectly to continue to talk about, instead of a cyanobacterial species, we're going to be looking at Prymnesium parvum, which is a eukaryotic, one of those eukaryotic algae. And so I have some microscopy images of Prymnesium here, and you can see, sorry, I'm just cleaning up my screen a little bit. Okay, and so you can see that they are eukaryotic cells. They have two chloroplast organelles. They've got a nucleus. They have two flagella for swimming around, and then this third appendage here, which is their haptonema. And the thing that people think the haptonema might be used for in these Prymnesium and related species of algae, which are called haptophytes, is they think that they might be involved in adhering to surfaces, but also involved in prey capture. And when you hear that, you may wonder for a minute, what do you mean prey capture? I thought we were talking about algae, and we are. So Prymnesium is a mixotrophic species, meaning it is both obligately photosynthetic, but it also goes around eating other cell debris and other cells in its environment. And so I have a video here of several cells of Prymnesium actively engulfing, phagocytosing and engulfing what used to be a green alga here, and this one taking a nice big bite, having a large food vacuole. And I have another video to show you just how I consider Prymnesium a photosynthetic predator in the environment. There's a green alga here, a poor green alga right here in a field of otherwise mostly Prymnesium. And you see that there's some sort of cell signaling or something that the other Prymnesium are recognizing, and it's causing them to swarm around this green alga and kill it and eat it. And I love this quote from Ralph Lewin, who's a famous phycologist. And he has this quote talking about people postulated that the heptonema might be involved in feeding, but many of us discounted this idea because haptophytes are generally photosynthetic. Little did we suspect what dark deeds could be done by such little plants. And so it's this behavior and considering what impact mixotrophy might have on Prymnesium that first drew me to working with this species. And what we noticed early on was that we saw a lot of variation in the feeding behavior of Prymnesium. So I'm looking at 4 different strains here and comparing how fast these strains are growing when you co-culture them with prey versus when you just grow them autotrophically by themselves. And you see that 3 of the strains above, if they're growing above the 0 line here, they're getting a growth boost when you co-culture them with something to eat. Versus 12B1, this light blue strain at the bottom, you co-culture it with prey species, and it doesn't seem to treat these species like prey. It actually seems to grow slower, as if it's growing in the presence of a competitor rather than a food source. And so some of the questions that I'm interested in my lab are, what are the genetic differences that are giving rise to this feeding variation and other variations in their toxicity, which we'll talk about in a second. And then how do these differences impact other aspects of Prymnesium parvum, including the species' ability to form toxic blooms? And so, yes, Prymnesium is a really important important species that causes these catastrophic algal blooms. This is a photo from the 2022 Oder River catastrophe in Germany and Poland. There was a massive bloom. It spanned the entire length of the river, which was the second largest river in Poland, caused 249 tons of dead fish, and that's an underestimate. And reports that came out following the bloom reported that the Prymnesium parvum bloom was really just a matter of time in this waterway, because the Oder River is permanently saline due to drought and runoff from agriculture and mining. And so these things, Prymnesium needs a little bit of salinity to bloom. It can't grow or bloom in strictly fresh water, but a little bit of salinity, even something that's just 1% what you would find in a normal marine environment, is enough for Prymnesium to get a foothold and bloom. And so the Great Lakes Aquatic Non-Indigenous Species Information System has listed Prymnesium parvum as a species that has the potential for high environmental impact and socioeconomic impacts if introduced into the Great Lakes for this reason. So where is Prymnesium parvum currently found in the US? A first documented case of a bloom of Prymnesium occurred in 1985 in Texas. And since then, it has been a recurrent problem in Texas and in nearby southwestern states. It blooms nearly every year. If you fast forward to 2016, Prymnesium is now detected in 23 states and counting. And there are quite a few factors that are associated with this increased incidence in Prymnesium blooms, including things like higher salinity in the water, which we already talked about, decreased wetland cover, increased use of glyphosate herbicides. But there's a lot about Prymnesium that we still do not know. And one of those things is its life cycle. So Barry mentioned that the cyanobacteria grow via just normal cell division. They don't have a sexual cycle. Prymnesium presumably does have some sort of sexual cycle, but we don't understand it, and we haven't documented it currently. So there's a lot there, and that would influence our ability to understand Prymnesium's population dynamics and evolution if we understood its life cycle. We also don't understand its genetic diversity. So how much genetic diversity is there contributing to the variation we see between different strains, and how much variation do we see kind of worldwide versus within the US or within Texas and within a single bloom? These were questions that my lab was really interested in addressing. And so Prymnesium parvum produces a suite of toxic compounds, but the most well-known of which are these prymnesins, which you can see are large, complex secondary metabolites that They have A, B, and C type Prymnesiums that are differentiated by the amount of carbon in their backbone skeleton. So these A type Prymnesiums have 91 carbons, B types have 85, and C types, which haven't been structurally characterized yet, have 83 carbons. And what's really interesting is if you look at the phylogenetic tree for Prymnesium parvum, you see that the different strains that are making the different types of Prymnesiums are evolutionarily related. So all the B-type Prymnesiums are made by Prymnesiums that are grouped together in a clade here, the C-types grouped together here, and I've expanded the A-type Prymnesiums producers because it includes Utex 2797, which is a really well-studied strain that was first isolated from Texas in 2001. And so when we were first getting into this project and looking for a strain to select for whole genome sequencing, Utex was an obvious choice, considering that it was a strain from Texas where blooms are most severe in the US and has been included in a lot of common growth and physiology experiments of Prymnesium. So we set about figuring out what we could about this strain and getting some background information on it for whole genome sequencing. And we determined that its genome size was around 274 megabases, which, just to put that kind of into perspective for you, that is a large genome, but well, kind of a normal average size genome for a eukaryote. So here is Prymnesium kind of in between our mottled fly Drosophila and some of our model plants, smaller than humans and our related primates, but much bigger than the cyanobacteria shown down here at about 4 megabases. So that all seemed very doable, but we had one big problem, which we noticed right away, was that Prymnesium, this Utex 2797, appeared to have really high heterozygosity, which that you can see by this secondary peak here in this Kamer plot on the left. This is telling us that Prymnesium has 2 copies of its gene. This strain has 2 copies of its genome, and that the 2 copies are very different from each other. And that's what's giving us this really high peak right here. And that can complicate genome sequencing and assembly. So we were a little concerned about that, but it also suggested that something interesting might be going on. And so we kind of filed that away for looking at later. Around the same time, we were also profiling an additional strain, 12B1, which was also isolated from Texas from a bloom in 2010. And you'll notice that its estimated DNA content was less than half that of Utex 2797, and with no observable heterozygosity. So this is suggested to us that 12B1 was a haploid. It had 1 copy of its genome, which made it ideal for whole genome sequencing. But we didn't want to get rid of Utex 2797 altogether because, like I said, it's so commonly used in the field. So we decided to sequence both strains using a combination of Illumina, Oxford Nanopore, and Hi-C sequencing technology. And I don't have time to go into the details of the assembly methods, but what we got out of it were really highly contiguous assemblies that are close, if not at, whole chromosome level assemblies. So the 12B1 assembly was around 100 megabases on 34 scaffolds, which I'm showing you right here. And the triangles are telling you that these scaffolds have telomeres on the end. So these could correspond to actual whole chromosomes of the 12B1 genome. And about 23,000, 24,000 protein coding genes. And if you contrasted that with Utex 2797, you basically see double of everything. Twice as big a genome, twice as many scaffolds, and twice as many protein coding genes, which really just further emphasized that something interesting might be going on with this strain, and something interesting about its evolutionary history. And in order to get to the bottom of that, we had to actually sequence more strains for some phylogenetic evolutionary context. So we selected an additional 13 strains from strains that spanned the diversity of the A, B, and C type Prymnesium producers. And what I'm showing you here, we sequenced these additional strains with Illumina. So it's much more fragmented assemblies, but good enough to get us predicted protein coding genes. And we used those 2,699 single copy genes to get a really well-resolved evolutionary tree for Prymnesium parvum. And you can see, like what we saw in our earlier example, which was data from just a ribosomal DNA sequence, at the whole genome level, we do see the different A types grouped together, B types grouped together, C types grouped together in this phylogenetic tree. And if we look at the rate of sequence substitution, the amount of sequence difference we see between these different clades, you can see that in particular, if we compare C-type Prymnesium producers to the A and B types, this dark brown color here is telling us that there's quite a lot of substitutions comparing between the different clades compared to looking within, where we see very little amount of variation. You'll also notice that if we look within the C types, we have this this strain here, K0252, which is from Australia. It is grouping sister to all the other C types, and it has a lot of differences between its genome and the other Cs, kind of suggesting that the C type lineage may be more diverse within that clade as well. And so we further broke that up into 3 separate subclades. And then the last thing that I wanted to point out on this tree is this very long branch leading to UTEX 2797. So in our consensus phylogeny, 2797, that weird genome that we were looking at before, it's grouping next to the other strain from Texas, 12B1, but it's got this long branch saying that there's a lot of sequence differences between it and 12B1. And drilling into what was causing this long branch, what we actually found out was that UTEX 2797 was actually grouping in multiple places in the tree, depending on what gene we were looking at. And so I have just 2 examples to show you. So here in this gene tree 1, you see UTEX 2797 is grouping with 12B1, the strain from Texas, and CCMP 3037, which is a strain from Wyoming. So 2 US strains. But in another phylogeny, well, you can see UTEX 2797 grouping in a completely different place with these 2 strains from Europe, from Russia and England. And if we looked across all the genes in the genome and summed up how many times did we see UTEX grouping here with the US strains, which we called the A1 clade, and how many times did we see UTEX 2797 grouping with these European strains, which we called the A2 clade, we actually found a really 50-50 split. 31 scaffolds grouped with the A1 clade, 31 scaffolds grouped with the A2 clade, and just 4 scaffolds had uncertain placements. And so what that really suggests is that UTEX 2797 is a hybrid. So it has 2 phylogenetically distinct A-type subgenomes. And so similar to a lot of hybrids we see in plant genomes. So I have some photos of these brassicas like broccoli and cauliflower. These are related vegetables, have a similar hybridization event in their ancestral history. And so when we looked at some additional strains from Texas, we were able to see that this doesn't look to be like a one-off. Strain 12A1, which was isolated from the same time as we got 12B1, also looks to be a hybrid. So it doesn't look like UTEX 2797 was just a one-off weird event. This seems to be something that is more common than that. And if we look at the kind of what other sorts of diversity are we seeing in this lineage, we see overall variation in genome size. So here I'm just showing you the average genome size, or the estimated genome size in megabases for these different strains. And you see that the B-type strain in particular have genomes that are more than twice as large as the genomes in the A-type. And so all of these genomes are haploid sized estimates. So how much DNA is in one copy of their genome. And then the C types are a little variable. We have the C1 clade has a smaller size genomes, and the C2 and C3 seem to have slightly larger genomes. Not only that, we also found that strains could be had different numbers of copies of their genome per cell. So I already told you that strain 12B1 is a haploid with one copy of its genome, but we found other strains that appeared to have 2 or even 4, so tetraploid copies of its genome per cell. And so this again is kind of similar to things that we see in plant genomes, like the strawberry. Strawberries are often polyploid, having multiple copies of their genome in a cell. And all of this taken together indicates just really unexpected and considerable levels of genetic variation, suggestive of at least 3 different cryptic species. Like at the very least, A, B, and C types appear to be 3 separate species, but there's arguments you could be made that the C1 lineage looks to be a separate species from the C2 and C3. Are the A1 and A2 clades here separate species, and are the hybrids that we're pulling out in our environment? Do these represent, was this an ancestral hybridization event and that give rise to a new lineage, essentially a new species? Or are these hybridization events something that kind of happen repeatedly and have happened multiple times and represent kind of evolutionary dead ends? These are things that we don't know the answer to yet, but it's really, really exciting. So what are the ecological consequences of all of this genetic variation? So let's bring it back to that graph I showed you earlier, looking at how these different strains prefer to grow with or without prey. And now we can look at these strains in the context of this phylogenetic tree and what we've learned thus far. And now I can tell you that these 3 strains here that seem to be growing faster when given prey to eat are all A-type hybrids. I don't have 8020 or 13A5 in the phylogeny, but I have data to back it up that all 3 of these are hybrid species versus the 12B1 is our A1 haploid-like strain. Not like strain, a haploid strain. And we also see that all this cryptic diversity has implications for the biochemical studies for Prymnesium. So if we look at the accessory genome of all of these strains in our analysis, and we just focus on genes that are variably present, so they're present in some strains and not others, that actually represents 44% of all gene families that we looked at. So a lot of the genes in this analysis are variable, present in some strains and not others. And the gene families that are showing up in this accessory genome are enriched in things like secondary metabolism and toxin biosynthesis. So there's the genes that are giving rise to the differences in the A, B, and C type Prymnesiums are in this accessory part of the genome, and we need to start. Another goal that we have in the lab is to see if we can identify these genes that are responsible for this, either the ecological or the biochemical differences between these different strains. Okay, so I think I have some more time, so I'm going to tell you a little bit more about some of our ongoing work, taking what we'd learned about the backbone phylogeny for Prymnesium and then going back to Texas and saying, what are we seeing in current blooms in Texas? We looked at blooms from 2018 through 2020. We looked at 9 blooms total, and we pulled out and we tried to isolate as many additional cultures as we could. And what we found, all of the new isolates from these recent blooms are bolded, and they're drawn in the phylogeny that I'd been showing you before. So what you see is that we've pulled out A1 type strains, we have A-type hybrids, and we have a lot of B types, which was really interesting to us because actually 8 of the 9 blooms that we surveyed contained these B-type Prymnesium parvum, which is the first documented case of B types being present in inland US. Previously, they had only been found in Europe, like that Oder River bloom that I was telling you about before. Those were B types, and along the Atlantic coast of the US, B types have been pulled out there. But this is the first time we've seen B types in Texas. And not only that, when we looked in single bloom, so I pulled out this 2020 bloom from the Possum Kingdom Reservoir, to focus on specifically, within a single bloom, within a 50 mil sample of this bloom, we pulled out A1 type Prymnesium, A type hybrids, and several B type isolates. So there's a lot, these cryptic species of Prymnesium are inhabiting the same bodies of water, and they're co-occurring in blooms, which is really exciting to discover. And so what we're starting to think of Prymnesium as now are diverse assemblages. They're not just a monoculture of one strain of Prymnesium that is exploding in the environment and taking over. There's genetically diverse strains of Prymnesium present in blooms, and that these different strains show a continuum of growth strategy preferences. Some prefer to grow autotrophically, some are more toxic and more mixotrophic, heterotrophic. The other thing, the final piece of this complicated environmental situation I have to mention with a little bit of time I have remaining is that these other species in the environment show variation to susceptibility to Prymnesium. And so what I mean by that, if you look at different strains of algae and you co-culture them with Prymnesium, some of them, even if you like this Denaliella and Phaeodactylum, if you co-culture them with just 1% of the culture is Prymnesium, 99% of the culture are these other two species, this prey species will quickly die. And within 200 hours of co-culturing, the culture will be entirely Prymnesium. Versus Kirschneriella, which is a green alga, even if the culture is 50-50 Prymnesium Kirschneriella, they appear, this species or strain of algae that we were working with appear entirely resistant to Prymnesium, which is another complicating factor that we have to take into account. And so what I think we have critical need for going forward are more studies looking at the relative abundances of all of this different genetic diversity, both Prymnesium parvum, cryptic species diversity, and the diversity of the other aquatic community members, both before, during, and after toxic blooms. And I think that will be really informative information to help us better understand when certain environments may be particularly prone to Prymnesium blooms, which may be more resistant to blooms. And that's the sort of, that's the thing that my lab is looking to do next. So stay tuned, and maybe in a few years I can give you an update on what we find out. So with that, I will thank the members of my lab. All of these people, all of these graduate students, postdocs, and undergraduates have contributed in some way to the project, as well as my collaborators and the NSF for funding this work thus far. And most of the stuff I was telling you about, you can get a full story of our genome sequencing and comparative genomics in our recent paper that came out in Current Biology. So I encourage you all to check that out. And that's it. I'm happy to take any questions.
All right, thank you. That was great. We want to return to a couple questions from Dr. Rosen's talk, and then there are some questions for you as well, in addition to all the applause. Hopefully you can see that there. The most common question from the first talk was, how many among the most common hab-forming species are nitrogen fixers?
Well, let me list off some that are not. No species of Microcystis, Planktothrix, there's Limnorhynchus. They do not have the capability of fixing nitrogen. They have no heterocytes. The ones that can fix it are anyone with a heterocyte. So you have the Dilocospermums, Raphidiopsis, aka Cylindrospermopsis, Cuspidothrix, Usmechia, which has been renamed from Chrysosporum, Phanozomenon. So anyone that's got a heterocyte has the ability to fix atmospheric nitrogen.
Okay, great, thank you. And then one more question. Does the presence of the gas vesicles reflect common ancestry, or has it evolved independently in multiple taxa?
We think that the gas vesicle, because you do find them in regular heterotrophic bacteria. So I wouldn't be surprised that it branched off early on, and cyanobacteria have taken it to probably a greater degree. But there are gas vesicles and heterotrophic bacteria.
Okay, great. Let's move to some questions for Dr. Wisecaver. So first question a lot of people had was about the glyphosate herbicides that you mentioned and how those affect the blooms of pea parvum.
Yeah, that's a great question and one that I don't know the answer to, but there's a couple of things that people have proposed. One is just that Prymnesium is resistant to the glyphosate herbicides, and so then by killing off a lot of things that would otherwise be competitors in the environment, it makes it opens the door for Prymnesium to bloom. There is some evidence, I think, that Prymnesium can use glyphosate as a source of phosphorus, but I don't know if that's really like biologically meaningful. Like how much phosphorus are they actually going to get in a real world situation from glyphosate? I don't know, but that's so kind of the two suggested things, either just inhibiting the growth of competitors and possibly as a source of nutrients, which would be pretty weird.
Okay. So what are the theories on why pea parvum produces toxins?
That's a good question. And so there are lots of different hypotheses that have been put forward. Given what we know about Prymnesium as a little microscopic predator, the theory that, or the thing that I find most compelling is that it's somehow using its toxin to capture prey. There is evidence that or when we watch Prymnesium under the microscope, it uses the haptonema. It kind of pokes at prey cells from its with its anterior side. The cell lyses. The toxin potentially causes the cell to lyse, and then Prymnesium will turn around and phagocytose up cell debris from its posterior side. But other people have put forward other suggestions as far as potentially being used again to inhibit the growth of competitors in the environment. I think we have to keep looking at, or like Barry said, they're not making these toxins to kill fish. So we need to look at cell level benefits for why Prymnesium is making these toxins. So things that, what's the benefit at low cell densities for making these toxins? I think capturing prey is one possibility. I don't know. There's sometimes I wonder if some of these toxins are, like Barry said, maybe these aren't toxic for like an ecological sense, but more for some sort of storage molecule or something that we don't fully understand yet. But I try to keep a really open mind for why Prymnesium is making the toxins, and we definitely don't fully understand that yet.
All right. Has the ability to detect P. parvum improved in more recent years? You referenced that there's more occurrences being mentioned. Do you think any of that's due to detection?
I do. I think that we have we are now detecting Prymnesium when it's non-blooming. So people are recognizing it in the environment and documenting it, even when it's not in a bloom state. So there could be some what was the specific question? Yes, we are detecting it more. Does that answer the question?
Yes, I think so. I think they're wondering if that was a factor in, are we getting better at detecting it? And thus that's why, part of why there's more that we're seeing. So
it seems to be both. I don't think it's just better detection. I do think that blooms are increasing in some areas, particularly in Texas. They kind of wax and wane. So they'll have periods where they'll have blooms every season, every year, multiple years in a row, and then they'll have some years like this year where it's a little quiet. So I think it's both.
okay, great. And then from an evolutionary perspective, over what time scale can new strains of P. parvum arise?
I don't know. I think that's a really good question. And something I would like to answer. But I do not know. So one of the things I couldn't help but notice for our A-type strains is that for the hybrids, we only have 2 strains of the A1 and the A2, but the A1s are in the US and the A2s are in Europe. Your mind can't help but wonder if there was a recent introduction of A2 strains into US that and this hybrid formed very recently. But I don't know the answer to that yet. And what we really need is we need more sampling. We need more than just 2 strains from each of these, better sampling to answer that question.
Okay, great. Got just a minute left, so we might have to leave the last questions, but we do have the speakers' emails on the slide here. So if anybody wants to reach out to them directly, please go ahead and do that. Yes, we are recording this presentation and it will be posted online. So if you want to share it with anyone afterwards, please go ahead and do that. And then we have another talk coming up as part of the North Central Region Water Network next week on creating resilient shorelines. So go ahead and check that out if you're interested. And I think that's it, unless we've got anything else we need to add last minute here.